Assalamualaikum warahmatullahi wabarakatuh. Hello everyone, let me introduce myself. My name is Naufal Arrafi Trisnanda. I'm from Biology Study Program at UIN Maulana Malik Ibrahim Malang. And I would to present my experiment on the effect of two factors at mung beans germination. These two factors is visible light and red onion extract as a natural plant growth regulator. This experiment, in accordance with the word of Allah Subhanahu wa Ta'ala in Surah Al-An'am, verse 95. A'udzu billahi minasyaitonir rajim. Bismillahirrahmanirrahim. Innallaha falikul habbi wan nawa. Yukhrijul hayya minal mayyiti wa mukhrijul mayyiti minal hayy. Dzalikumullahu fa anna tufakun. The translation is, it is God who splits the grain and the seed. He brings the living from the dead, and he brings the dead from the living. Such is God, so how could you deviate? This experiment as a requirement to take a part in the semi-final of the IWSC Biology Olympiad competition. Okay, now I will show you the material used in this experiment.
From material I have red onion, mung bean, cocoa bean, some water and tissue. Tools that I used are filter, blender, petri dish, measure glass, pincet, knife, germination tray, spray bottle, millimeter block, caliper, and digital scales. There are 4 method stages as follows. Let's discuss it. Peel and remove 500 gram red onions cut, then grind it with blender until smooth like this. Put grounded red onion at measure glass, then pour water until 200 ml. Filter red onion soaked water to remove red onion residue, so I get the red onion extract liquid. This is first factory treatment at mung beans. That is soak mung beans in water and red onion extract at 3 minutes. Then take and put mung beans at petri dish. Most component of this media is from cocoa peat. Coconut peat is made from coconut husk that has been ground into powder. Put enough cocoa peat powder at 6 germination tray, then spray with water until look moist. Put 30 green beans that have been soaked into each medium with 3 trays for water treatment and 3 for onion extract treatment. For placement, it is arranged 5 columns by 6 rows like that.
After that, put tray containing mung beans in a shady place, then close trays with tray cover to let the seeds germinate, particularly until its radicals grow. After mung bean radicals grow, move these trays into each level of the germinate rack. Each level of this rack have differ in the visible light spectrum, that are green, blue, and red. It aims to treat these mung beans with visible light factor. Then cover entire rack with black curtains to block other light. And then we wait it germinates completely for 3 days. Take mung bean sprouts to measure height from primary leaf tip to the part of radicle above media. Use caliper.
At mung beans germination, there are many parameters for observe effect of seed treatment. For this experiment, I took sprout height for first parameter and growing root length for second parameter.
Then measure height by digital scale.
And the last parameter is seed germinability, known as count sprouts from a treatment that grow normally.
After I collected data on these 4 parameters from 10 sprout samples from each treatment with visible light and red onion extract or water, I got data on sprout length, sprout weight, and seed germinability. Then I analyzed the data, use SPSS program to do the ANOVA statistical test, and 5% DMRT for the test. And the results as follows. This analysis result is because red onion extract contains natural plant growth regulator or natural PGR known as auxin and rhizocalin, which stimulate radical growth, so can support root growth. Also, auxin substance stimulates cell division, so boosts elongation of epicotyl, hypocotyl, and rising plumels. Eventually, this impact is influenced length and width of sprout. In other words, red onion also contains flavonoids, which can protect seed from oxidative damage during germination and pathogen infection, so can increase seed viability and germinability. Also, the saponin content in red onion can increase water absorption by the seeds. The onion extract treatment is supported by visible light treatment in the red spectrum. Red light spectrum is the most important spectrum in regulating the growth of seed plants, especially in early phase of plant growth, because it can increase the germination rate and hypocotyl elongation in mountain sprout. Red light spectrum play a role in activate phytochromes, which are photoreceptor pigments that are important in the photosynthetic process of germination. Red light also increases chlorophyll biosynthesis rate and the activity of stores hydrolyzing enzymes in cotyledons to support the mountain germination. Several studies show mountain sprouts that have been treated by red light at various exposure duration had the highest biomass and sprout length compared with other light spectrum such as blue and green spectrum.
That's all my speech. I do hope that my speech will be useful for us. If you found many mistakes in my speech, please forgive me. The last essay. Wassalamualaikum warahmatullahi wabarakatuh.